Hey, today we're going to talk about what goes in an introduction section. Now, a lot of people think they know what goes in an introduction section, but when I read what students write for their introductions, I'd like to think that sometimes people don't stop and consider all of the things that need to go into the very first section of a paper. So let's talk about that. So a lot of introduction sections are just basically one big blob of text. So a lot of people don't realize that they can be broken down into three main parts. The first part is talking about why the subject that you're studying is important. In other words, why does it matter? Why should people care? What importance is it in the environment if you're doing something environmentally related? From there, you get into the part that most people think of as the literature search part, where you're going through a lot of what's been done in the past. And essentially what you're trying to do here is you're trying to give the reader all the background theory that they need to understand the third part of your introduction section. And the third part is a brief statement about what it was you were trying to find out when you did the study. And why were you trying to do it? It's very helpful before you actually start writing an introduction section to go through introduction sections that other people have written and look for these three major parts. So what I'm going to do here is I'm going to talk about the strategies that people take as they write an introduction section. And I'm also going to walk you through the introduction section of two different papers, which happen to be the papers that we talked about when we were looking at method sections. So let's talk about the approach you should take when you're writing your introduction section. In my mind, you may be different, but to my mind, the easiest part of the introduction to write is the first part. The paragraph where you're talking about why your topic is important, why it's relevant to the scientific community, and possibly even why it's important to society. So if you've done a proposal, you might tie in a little bit from your broader impacts. The reason this is the easiest is because you probably already know why this is important. That's why you chose to do the study in the first place. But here's the thing, you can't just tell people why you think it's important. You have to justify its importance using previous studies. And so one of the mistakes that I see when I'm reading student drafts is that they save all of their citations for the second part, the lit search part of the paper, and they don't put them in the relevance paragraph. So make sure that you come up with good peer-reviewed articles that emphasize why this is important in your field of study. Now, you might be tempted to just start at the beginning of your introduction and write from beginning to end, but I highly recommend that the next thing you write be the last paragraph, the one where you explain your motivation to do the specific work that you actually did. And what you need to do is be very clear in explaining specifically what about this project has not been done before. So, for example, you could be analyzing something about the chemistry of the Hood Canal. And maybe no one has analyzed that part of the chemistry of the Hood Canal before. Maybe they've looked at other components of the chemistry of the Hood Canal. Maybe they've measured the thing you're going to measure, but somewhere else in Puget Sound, not in the Hood Canal, right? So people might have done something that's similar, but not quite the same. Now, sometimes they've done exactly what you're doing, but it is something that might change over time and no one has measured it for a good long while. That is also a justification for doing your study. So you want to explain what has not yet been done and why you measure that thing that has not yet been done. Essentially, what you're doing here is you're writing your research questions, and in a poster, if you remember what I put in my poster video earlier, you would actually write them as research questions, like how high are hydrogen peroxide levels in the oligotrophic ocean? And you might also put those in an oral presentation framed as questions. But in a paper, you don't frame it as a question. You just explicitly state, this is the thing that we were studying, and the reason we were studying it is because no one has specifically studied this before. So once you've done that, then you can go back and you can write your literature search. And the reason I suggest that you write your lit search last is because writing the beginning and the end help you to frame what goes in the lit search section. So I've mentioned before that I've read a lot of student drafts, and one of the things I see in student drafts is a lit search section that is very, very sparse. In other words, there's only maybe a paper or two cited there, not very much. And when I talk to students about this, they say, yeah, but there was nothing that was relevant. And when I push further, what usually happens is that they tell me that, well, no one has done this exact thing before, therefore I can't find papers on it. And that's not the point of the lit search section at all. Yes, we know that no one has done this thing before. That's why you're doing it. So what can you put in? So if you're doing an environmental paper, as many of my students are, one of the things that you'll definitely want to address is what is known about your field site. Some people will put this first, some people will put it last. It depends on how you're framing your research questions. So what has been done on your field site? If anything has been done on your field site. Now, what if nothing has been done on your field site? What if you're working in an area that people just don't study or don't publish research about? Well, then what you can do is you can look for similar field sites that you can use as an analog for your own field site. So, for example, if you're looking for research on Bud Inlet or Eld Inlet, which are the two inlets of Puget Sound right by our campus, you might not find very much. And then you'd have to look and say, well, okay, what else is near here that might be like here? And so what you might do is you might look for studies on the Nisqually, because of course they did a bunch of research before they restored the Nisqually Delta. Okay, so that's thinking about your field site. Now let's think about what it is that you're actually analyzing. So if you're doing chemistry, and of course that's always the thing I think of first is, doing geochemical analysis, you might want to be focusing in on one particular thing. Sure, you might take a whole bunch of relevant measurements, like you would probably measure the temperature and stuff like that, but your paper is probably going to be focused on one or two specific analytes. And so you want to put in any research you have about how your analyte behaves in the sort of environment you're dealing with. So for example, if I were looking at Bud Inlet or Eld Inlet, I would want to look at what people had measured in estuaries. Because whatever I'm measuring, it's probably going to be different in an estuary than it is out in the open ocean. And it certainly is going to be different than what I see in freshwater. So what is the closest thing I can find to what I want to measure? Now, if you're doing biology, what you want to think about is what sort of related research is there about the specific organisms that you're trying to study. Okay, so let's say you're looking at jellyfish in Bud Inlet. Okay, what sort of research has been done on jellyfish? Be specific. What sort of research has been done on jellyfish in estuaries? Now let's say that you have magically discovered a new species of jellyfish. No one's ever written about that jellyfish before. That's okay. You can look for research on related organisms. In this case, that would be other jellyfish that live in the same area. So you could do the same thing for diatoms. No one's ever measured this in diatoms before. What can I do? Well, what sort of other phytoplankton have people measured this in? That may be the closest you can get. People have never measured this in any phytoplankton before. Great. Are there any other sorts of microbial life that they've measured this in? Right? So you're always going for as close as possible to what you're studying. And if there's nothing that's super close, get as close as you can. That's the important thing. Because what you want to do is you want to come up with a hypothesis. The hypothesis may not go in your paper. In fact, it frequently doesn't. If you read an introduction section, there's frequently not a very clearly spelled out hypothesis. But often when you're setting up your experiments, you want to know approximately what you're looking at. So if you're thinking about measuring something, you want to have an idea of what range that something might be in, because otherwise you could measure a range below or above that range. You might not even see it. So you always want to have an idea, if you can, about what might be going on. And so you're always going to look for the most relevant literature, whatever that happens to be. And if you get stuck thinking about, well, what could be relevant to what I'm doing, talk to one of your professors, because we might be able to help you with that. That's a quick overview of what goes in an introduction. Now let's go back and let's look at two of the papers that we looked at earlier. So we're going to start with the Rowe 2012 paper, The Acquisition of Iron by Trichodesmium. And this is up on Canvas in the week 2 module, in case you're looking for it. So let's look at the first paragraph in the introduction. Iron is an important micronutrient for nearly all organisms. Notice how they start straight out by saying important. Why is it important? Now they go into detail. Notice that they're not just saying it's important, they're backing that up with specific facts. It is used in diverse biological processes, including nitrogen fixation, photosynthesis, DNA biosynthesis, and electron transfer. And there are your references. Although iron is an abundant element on Earth, iron is scarce in the surface ocean and has been identified as a limiting micronutrient to phytoplankton. And here they cite a review paper which ties together all sorts of research papers into one concise summary. And so you can go through, you can continue down, but let's just look at this last sentence. And it says, because of the extremely low concentration of inorganic iron in surface seawater, it is believed that iron ligand complexes in seawater are likely important sources of iron to marine microorganisms. So they started with a very general statement of importance. Iron is important. And now they're talking about what specifically is important about iron, which is that in order for marine microorganisms to use iron, they need iron ligand complexes. So that is the important statement. Now, the second paragraph, remember, is the literature search, where they talk about all the things that have been done in the past. So they talk about iron limitation in a number of different things. For example, Prochlorococcus, Trichodesmium, reference. Why is Trichodesmium important? Reference. More Trichodesmium stuff, reference. Now you notice they're focusing a lot on Trichodesmium, and that tells you that Trichodesmium is going to be important in the study. But now they're talking about heterotrophic bacteria. Can produce siderophores under iron-limiting conditions. But only a few cyanobacteria, reference. Okay, and so you can see this is a really long paragraph. There's 2 paragraphs. And basically, each sentence or two is a summation of what has been done in regards to iron, Trichodesmium, and heterotrophic bacteria. Now, this final paragraph talks about what they're going to do. And you'll notice that part of this is still sort of lit searchy. It's kind of transitioning from the lit search into what they're going to do. So they're starting out by saying, hey, let's talk about a few things that we know about trichodesmium, mainly that trichodesmium forms colonies, and there are a whole bunch of other things that live with these colonies. Bacteria, dinoflagellates, amoebae, and et cetera, and they are giving, once again, they're giving references. So now what they're saying is, because these colonies are known to have very high densities of bacteria that are associated with them, that means they also need a lot of iron. And then right here, you can see there's the very last sentence that is very explicitly spelling out what they're doing. This study employs laboratory experiments with representative organisms to more closely examine potential mechanisms of iron acquisition and cycling within trichodesmium colonies. Now, if you look at that last sentence and then you go back and you look at all of the other sentences in that paragraph, you will see how they support that particular statement. Representative organisms, which are associated with trichodesmium colonies, well, that whole paragraph talked about organisms associated with trichodesmium colonies, and iron acquisition. Now, the other thing, if you go back and you look at that paragraph, they never explicitly say, hey, no one's ever really looked at iron uptake in trichodesmium colonies, but there are hints where they say only a few bacteria have been observed. Trichodesmium has not been shown to produce, so you can go back to the literature search. Everything in the lit search section, as well as that last paragraph, is tied into what you need to know before understanding why you would study specifically potential mechanisms of iron acquisition and cycling within trichodesmium colonies. And now let's look at the Lich paper. Now, this paper is a little different in that the introduction is actually broken down into 3 sections. There's the first part that says 1, introduction. And then if you scroll down, you'll see section 1.1, background, section 1.2, motivation. This is also a pretty long introduction. In this particular case, the sections are more or less broken down along the lines of the 3 sections that I talked about earlier. So the first part that just says introduction is really the importance part. So you can see where we start out, Martian Paleolake basins are prime habitability targets for future Mars rover missions. So there's something important. We are sending expensive spacecraft to Mars to look for life. We ought to know where to send them. Life on Earth mostly began in the deep Hadean Ocean, where there were significant geochemical gradients for energy, substrate catalysts, etc., okay? So they are talking about, where did life start? Where do we look for life? These are fundamental research questions that we are still trying to figure out. And that's the first paragraph. The second paragraph, in very simple terms, says, hey, it's really hard to get to Mars, so let's start looking at places on Earth that are very much like what we expect Mars to be. You'll notice that this very basic introduction, there's a little sentence right at the very end that says, here's what we're going to do in the study. So you might think, oh, that's the statement of what we're going to do. And it kind of is, but you'll also see, okay, 1.1, background. Now here, what we're going to do is we're going to look at everything that's important for this study. The first 3 paragraphs, if you go through, talk about paleolakes on Mars. And you'll notice that there are numerous references here. And then in paragraph 4, right at the bottom of page 600, now they say on Earth, examples of basically paleolakes, and this is what those are like. Why do we think they're useful as analogs for Mars? Now we get into the motivation section. Here we're talking about why we're doing this study. Why are we doing this study? Start off, we've identified paleolakes on Mars, but we also are not 100% sure how consistent they are with the lacustrine sediments. Okay, so we've seen pictures from afar, but we've never actually been there to see if our remote sensing spectrometry is actually indicative of what's there. And you can see here's a whole bunch of information on complex mineralogy, terrestrial analogs, And then here's the final statement. This is what we were working up to. In this study, we present the Great Salt Lake Desert as a valuable terrestrial analog. Okay, so we kind of said that earlier in part 1, but here we're being very specific about how we talk about this as an analog. And all of this stuff ties back into those paragraphs and paragraphs before it, where we're deciding, why are we actually using this particular field site? Why do we even need to do this study? And what is all the background? Even tying back into the background paper, look back and see how everything that's in that section 1.1 is necessary for justifying these last 2 sentences. So that's a quick look at introductions. I'm not spending a lot of time on this because I know that many of you have written introductions. Be sure to check in with one of your professors if you need a little help figuring out what you can still add to your introduction section after you've done the initial draft. And I will see you soon. We'll talk about results section.